Good evening, and welcome to Monobiology. This evening we're going to talk a little bit about cloning. Cloning actually occurs all the time in nature. In fact, twins are nature's clones. And it doesn't just happen in animals. It happens all the time in yeast cells and even in Hydra. Of course, bacteria also reproduce asexually. Any type of asexual reproduction will produce a clone. A clone simply means a genetically identical cell. Once people understood a little bit more about genetics, people thought, hey, why don't we try and do this cloning thing on purpose? They initially started with plants, taking cuttings from plants, from trees, planting them, and watching them grow. Of course, those were clones too because they were genetically identical to the parent. Eventually, people got a little bit more ambitious and thought, if we can do it with plants, and if bacteria and other single-celled organisms can do it just on their own, then why can't we do it? So we set out to do it. The initial experiments were done with stem cells. Stem cells are undifferentiated cells, and because they're undifferentiated, all of the genes in the chromosomes are still turned on. And so if you were to take the DNA from a stem cell and place it into another cell, you could, in theory, allow that to grow into a new organism. This is the basis of cloning on purpose. Initially, when people started performing cloning techniques on animals, they only used stem cells. That limited people in what they thought could happen, and the future of cloning was becoming narrow. A group of scientists in Scotland set out to prove that it was possible, in fact, to use cells other than stem cells. In other words, differentiated cells, like these mammary cells right here taken from this sheep, could be used to do exactly the same thing and clone a sheep. And eventually, through the process that we're about to discuss, Dolly the sheep was cloned in 1996. The process of this particular cloning method is very similar to the process outlined in the textbook. First, they take a sample of cells from a cell donor, which in this case happens to be the sheep right here. Next, they take an unfertilized egg from another donor. They remove the nucleus from the unfertilized egg. They remove the DNA from the mammary cells here. They insert the DNA from the mammary cells into the unfertilized egg. Now here's where something important happens. In order to stimulate the chromosomes within these differentiated nuclei, a current was used, and this current stimulated these cells to grow and divide. And once that happened, something miraculous occurred. They didn't just grow into mammary cells, they grew into a developing sheep. That developing sheep was then placed inside the surrogate ewe, which is a female sheep, for those of you that weren't aware of that. And several months later, out popped Dolly. So the process is very similar, but this was a very important thing to have proved. You can use cells that are not stem cells to make clones. Now, unfortunately, Dolly only lived for about 6 years. And there's some speculation, although it's never been completely proven, that because these cells had already undergone several several cell divisions, that Dolly would never live nearly as long as a stem cell would. Because remember, there's only a certain number of cell divisions a cell can go through. So these cells here would have gone through several cell divisions coming from an adult sheep here. So some people believe that Dolly died because these cells were already old, and therefore the chromosomes were already old. Other people don't think that's necessarily true. It remains to be seen, and hopefully in the future some of these processes will become a little bit easier to use, and they can have lots of benefits. Of course, there's always the downside of these things. Many religious groups feel that this is an evil technique, and that designing these particular processes only leads to people playing God and killing innocent organisms. Many people believe by the time conception occurs, which would be the fertilization of an egg, an organism is alive, and therefore if you destroy it, or if it dies in the process, then that could be considered murder. Just like some people believe that abortion is murder. On the other side, if this process was able to work on humans, then our identities would be in jeopardy, some people believe. Not our identities like, hi, I'm Evan Monahan, but my genetic identity. Imagine if somebody could clone me and then use my DNA to commit crimes or to do not so nice things in other parts of the world. That would not be a good thing, for sure. Certainly, probably you guys wouldn't want that to happen, somebody to be running around with your DNA. And some of these ethical issues have to be worked out before we start even attempting to clone humans. I hope this gives you a better understanding of the process of cloning, and certainly if you have any questions, feel free to bring them to class. Thank you.